Thank you so much for joining today's webinar presented by our STARR team. They're going to be showcasing their new Lago-X system that's now available in their core facility. My name is Cameron. I'm the manager of the Research and Innovation Core's operations team. We support all our core facilities in their efforts to keep bringing new instruments and technology to the research community. This session is really part of our ongoing effort to highlight those resources across the UHN core facilities. We have cores in flow cytometry, histopathology, genomics, and a whole bunch of other ones you can check out at our website. But for today, we're going to be talking about STARR and the Lago-X. So I will turn things over to Naz, who is the chief scientist and manager of the STARR facility. Thanks, Naz.
Thank you, Cameron, for the introduction, and good morning, everybody. And thank you all for joining our STARR's webinar. It's great to see the interest in STARR's new equipment. So for those who haven't heard much about STARR, the STARR Core, for the first time, I will provide a brief overview of our core facility. So we'll go on to the next slide. So STARR is a preclinical imaging and histology facility. There we go. We do provide services to all UHN internal and external academic scientists and researchers. STARR does provide service support to also our industry clients in their perspective in preclinical research studies. We are located on the 7th floor of the Mars building at the PMCRT tower. We've been supporting well over 1,800 researchers and scientists working very closely with STARR for coming close to 10 years now. And so STARR's core mission, as noted here, is that we empower researchers with innovative preclinical imaging technology, software, and collaborations to accelerate multidisciplinary translational research towards scientific advancement. I do want to highlight our preclinical imaging services at STARR, ranging from image-guided radiotherapy to nuclear imaging, radiopharmaceutical studies, to ultrasound and optical imaging, which is going to be the topic of our webinar this morning. So I'm very excited to introduce our new optical imaging equipment called the Lago-X, which will eventually replace the current 15-year-old Xenogen so-called IVIS system that most of you are very familiar with and have utilized for your studies at STARR. So the agenda for the webinar will be as follows. We will provide a brief overview of the in vivo optical imaging system, the features of the Lago-X, its applications, troubleshooting. We do have Permega, Simon Moe, on the line to provide a brief overview in the later half of the presentation on some of the luciferase substrates that some of you may find useful for your studies on the Lago-X system, as well as the Bruker scientists as well on call for any technical questions near the end. And of course, we will leave lots of times for questions for you all to ask any one of us on our team. And so I will now pass it along to Rita Chen, a STARR research technician leading the Lago-X launch. Over to you, Rita.
So thank you, Naz, for the introduction. We're just going to hop into our first topic, which is in vivo optical imaging. I'm just going to briefly go over how it works, its advantages, as well as its limitations. So for those of you who don't know, optical imaging is the detection of visible light photons from either bioluminescent or fluorescent probes. How the light is produced is different in bioluminescence versus fluorescence. So in bioluminescence, the light is produced when the luciferase enzyme converts the injected luciferin substrate into oxyluciferin. So this reaction produces a light, which is then captured by the camera. And then in fluorescence, imaging light is actually produced by a fluorophore being excited by an external light source. And so that excitation light will emit a light which is captured by the camera. So advantages of optical imaging, it has a high sensitivity and high throughput. And so bioluminescent imaging has been known to have a low signal to high signal to noise ratio, which makes it ideal for whole body in vivo animal imaging. It's fast and provides real-time data acquisition, and so acquisitions only take a couple of seconds or minutes compared to other imaging modalities. And it's also non-invasive or very minimally invasive, as very complex tools and procedures in order to get imaging results. Okay. So going to go over the limitations for optical imaging. As we all know, every imaging modality has its limitations. And so mainly it would be low tissue as well as a low spatial resolution due to light scatter, as it does rely on light emitted from these probes. And so fluorescence also has an added issue of autofluorescence. So just get my pointer here. So as you can see from this image here, it's an orthotopic model. It's an orthotopic cervix model where the tumor is labeled with DS red. You can see from these first two images here that there's actually a lot of autofluorescence from the mouse, particularly the fur regions here. Even after opening up the mouse, the fluorescence coming from the mouse itself is quite strong in comparison to the tumor signal. So only after covering it up do we see the fluorescent signal from the cervical tumor itself. So here's a photo of it X-size. You can see that it's showing a strong signal for DS red. And there is a secondary photo here that shows lymph node activation from the tumor. The second figure here is essentially showing the depth penetration for various fluorescent probes. DS red excites at a range of 560 nanometers. And so you can see from this diagram that it's actually very superficial. So the main limitation is tissue depth penetration. And so there have been a lot of efforts in order to kind of mitigate this. And so the solution would be to use NIR probes or redshifted luciferases. And so this first diagram here is essentially showing that hemoglobin is the main principal absorber in visible light, and water and other lipids are principal absorbers in the infrared light spectrum. And so within this near infrared window, all three of these things, blood, water, as well as lipids, have their lowest absorption coefficients. So this is approximately the 650 nanometer to 900 nanometer range. And so choosing probes within this range would not only maximize your tissue penetrance, but it'll also minimize the autofluorescence from non-targeted tissues. The second image here is for bioluminescence. And so it's comparing three different substrates. So there's acalumine, diluciferin, as well as cyclic luciferin. You can see their excitation peaks here. Acalumine having a 677 nanometer peak emission. You can see from the results here below that acalumine, the near infrared shifted bioluminescent probe, actually has a higher tissue penetration percentage in comparison to diluciferin as well as cyclic luciferin. So now moving on to features of the Lago-X, I wanted to particularly focus on their fluorescence excitation as well as their kinetics module. So just general features, going over it. The system holds up to 10 mice. It has a large field of view, 25 centimeters by 25 centimeters. It has a minimum detectable radiance of 45 photons per second. They have a multi-LED excitation filter wheel, which is located at the top, with 14 LED excitation wavelengths ranging from 360 nanometers to 805 nanometers, as well as 20 fluorescent emission filters. And so this is the only system in market, I believe, that uses these LEDs. They have a vertical door to minimize anesthesia exposure, as well as an air-cooled camera. It cools to an absolute negative 90 degrees Celsius temperature. It has a heated platform as well as an integrated X-ray camera on the bottom. I do want to point out that the system would use different gas than the other systems that we have on the floor. So we've changed the anesthesia from isoflurane to sevoflurane. So these gases are actually not interchangeable, so please don't mix them up when you are refilling our vaporizers. So I'm sure you guys are all wondering how the Lago-X compares to our existing 15-year-old Xenogen system. So I've made this comparison table. So as we're all very familiar with, the IVIS system images up to 5 mice at a time, whereas the Lago-X system has an improved capability of imaging up to 10 mice at a time. So it's double the amount of mice for a higher throughput system. The field of view ranges from 4x4 to 23x23 in the IVIS, whereas the LAGO ranges from 6x6 to 25x25. The IVIS system uses a single white light excitation for its fluorescence, whereas the Lago-X uses a multi-LED excitation filter wheel. Both systems are quite sensitive, but the Lago-X has a greater dynamic range. It also detects, it has a minimum MDR of 45, whereas the Lago-X has a MDR of 70. And of course, the IVIS system doesn't have integrated X-ray, whereas the Lago-X does have an integrated X-ray. So we're very excited about that new capability. So I wanted to talk a bit about the LED versus white light excitation for fluorescence and why LEDs are actually a little bit better. And so LEDs are more stable over time and have a longer shelf life compared to white lights. And there's also no warm-up time required. The spectrum is more precise in comparison to white light, whereas the spectrum is more variable, as you can see here. In order to kind of minimize the variability of the white light, you would need a warm-up time, ideally like 15 to 20 minutes. So the LEDs are able to excite at more exact wavelengths at a higher capacity compared to white light sources. So this graph here is also showing that you can have a greater signal intensity, which is directly proportional to the power setting on the LEDs, which is a setting that you can play with and interchange on the Lago-X. So the Lago-X does have a new software. It's called the Aura software. It is a license-free software in which you can request a download link from the vendor off of their website. I just want to take a moment to kind of explore the tools that you're all familiar with on the IVIS. And so the color range control for image adjustments, as well as optimizing signal is found on the right here. Your ROI measuring tools is on the side here. They have a few different imaging modes. They have a new one called Easy Mode, and it's essentially 3 easy clicks in order to get a bioluminescent or fluorescent acquisition. They have their classic mode, luminescence or fluorescence modules. They have just an X-ray module, group acquire for your protocols, or for your very specific protocols. And then they also have a new kinetics module, which I will touch on a little further. And then they have a user acquisition preferences set up here so that you can actually customize your profile to how you'd like. So moving on to bioluminescent kinetics. So as I'm not sure if you're all familiar, but when doing bioluminescent workflows, it's important to establish a kinetic curve in order to kind of figure out when the reaction hits its peak saturation point. And essentially you always want to image at this point for better reproducibility. And so going through the differences between the IVIS workflow, whereas the Lago-X workflow, with the existing Xenogen system, you would set up a time series study. It would take all these images, so you'd have to manually draw your ROIs and then get your ROI summary table, which you can then plot. And so you can either do this in 1 sitting or 2 sittings. You can plot the data, or you can kind of just look at your summary table and guesstimate approximately which time would be your peak saturation point based off of the maximum radiance that it provides. But in the Lago-X softwares and the new Aura software, it actually has a kinetics module to help you figure this point out a lot easier. And so it will generate a kinetic curve in real time, as well as provide a summary table of its ROI measurements. So you can see here, it's measuring this half of the mice. So it's imaging 10 mice. There would have been 2 graphs, but we're focusing on this graph. It plots RIs 1, 2, 3, 4, and 5. And this black line is essentially showing an average of the signals, and then it provides a summary table here as well as giving an average. So you can figure out where best your peak saturation point is. And in this case, it peaks at about 12 minutes. So now moving on to applications of the Lago-X system. A main application would be early cancer cell detection. You can monitor signal over time as well as your tumor growth. And so this image here is showing an orthotopic glioblastoma model where they have implanted cells in the left frontal lobe of rat brains. You can see from this figure that there is an increased signal over time. You can also see the signal getting larger as the tumor develops. Second application, or the same application, is I just want to focus on a bioluminescence with an X-ray overlay. And so X-rays have become a very popular method for providing anatomical context for bioluminescent as well as fluorescent signals. But because repeated X-ray exposure can cause DNA damage, upregulate radiation-sensitive proteins, induce cell stress, this paper kind of aimed to evaluate the potential damage in a pancreatic tumor model. And so they imaged these sets of mice for 10 consecutive days using bioluminescence imaging as well as a high energy X-ray. And so you can see this first image here shows the bioluminescent images for the pancreatic tumor models on day 7 and day 17 in the X-ray group versus the no X-ray group. And then the second image essentially shows the bioluminescent signal overlaid with X-ray as well as just the X-ray image on the bottom. The paper didn't find any significant damage impact from the X-rays compared to their non-X-ray groups. So another application is tumor targeting and tracking of nanoparticles across the blood-brain barrier. And so a common application of the Xenogen is for biodistribution tracking. So this paper looked at a dye, IR780, as it's known to have excellent tumor targeting as well as imaging potential. But due to its hydrophobicity, it kind of limits its properties in clinical use. And so the group has made these nanoparticles and micelle formulations to kind of facilitate the use of this IR780 dye. And so this figure shows fluorescent imaging of these malignant brain tumors at different time points. And so it was imaged at an excitation of 745 and emission of 780, and it was only exposed for 1 second. And so you can see that the R780 dye is targeting towards these brain tumors, as you can see from these fluorescent images. And at 2 points, 24 hours and 96 hours post-inoculation, do you see they took bioluminescent images, which is essentially just showing signal coming from the tumor. So you can see that there's a co-registration here that the fluorescence is targeting towards these tumors that they've put in, which have a luciferase expression. The last application I want to touch upon is treatment response. And so this paper used an orthotopic T-cell acute lymphoblastic leukemia xenograft model in mice. They did 2 injections into sublethally rated mice, and they randomized 20 mice into their 4 different treatment arms. The first one being vehicle, then AZD, then Pontonab, and a combination therapy of AZD and Pontonab. You can see from this figure here that the combination therapy had a lower tumor burden in comparison to the other 3 treatment arms that they had tested. So moving on to troubleshooting as well as frequently asked questions or frequently encountered problems. So the first one is anesthesia. I'm sure most of you are very familiar with this issue. It's not that common as long as you kind of remember to turn the flow on correctly to its proper ports, but it does happen to the best of us. And so in the case that your mouse wakes up due to anesthesia malfunction, you want to check that your connections are tight and that you actually have flow going towards the cabinet. So in the Lago-X system, the mouse manifold actually plugs into 2 ports. And so you just want to make sure that these connections are fully plugged in and tight. There is like an audible click. And so you just want to make sure that they have been fully clicked in. Otherwise, there is just isoflurane or sevoflurane essentially leaking into the cabinet, and you wouldn't get proper flow into the manifold. And the second issue is if your mouse does wake up and you do lose him in the cabinet, as you're all familiar with in the IVIS, it can fall underneath the cabinet. And so the Lago-X is the same problem. They have an under platform access button, which you can press, which will lift the stage all the way up. And then you can check under here for your mouse. The second place your mouse could be is actually because the door raises vertically. Just behind the door, there is like a very small ledge that your mouse could have gone into when the platform was moving. So that's a secondary place to check. Another common problem is not being able to initialize the system. And so this happens when the emergency stop button is accidentally pressed and you don't really notice. So as you can see from this picture here, the Lago-X does have the emergency stop button like right at the front. It is quite easy to bump into accidentally. And so if you do bump into it and you try to initialize the system, it's not going to work. And so to mitigate this, you're going to have to unlock the e-stop button so that it stops flashing. And then you'll have to complete a full power cycle in order to restore the communication between the system as well as the computer. Okay. The second one, which I don't really anticipate being an issue due to the high volume of Xenogen users that we already have, but it's communication issues between the computer as well as the system. And so when the system sits idly for a long period of time, they use USB connections. And so Windows likes to find a way to turn those USB connections off if they're not used very routinely. And so to kind of restore the communication, you just have to complete a full power cycle as well. The last issue, or another one is essentially having a PC freeze. This can occur when you continuously hit the cancel button on the software. And so Aura, when you start an initial acquisition, it likes to finish that acquisition even after you hit cancel. So it wants to complete what it's doing before terminating everything after you hit cancel. So repeatedly hitting cancel on the system isn't really going to help terminate it faster. It's just going to lead to the PC freezing or crashing out or the software lagging. And so normally when this happens, typically you would need to restart everything and potentially also perform a power cycle. But this problem can be totally avoided with patience. And so if you just need to cancel the workflow, just hit cancel once and hopefully you won't run into a PC freeze. And the last troubleshooting thing I'm going to touch upon is image optimization on the Lago-X. And so the control or the sliders on the software are actually quite sensitive. And so they do recommend that you just manually adjust or optimize your images. And so you do this by setting the color range threshold and minimum to 0 so that you can see both your strong and weak signals. You can then slowly slide the color range threshold bar up to a point where you've gotten rid of the background, but you've kept the weaker signals. And then you want to check what that number would be and change the color range minimum to match. And then optionally, you can change your color range maximum as well. Okay. And as has mentioned previously, we do have Promega on the call, namely Simon and Leah, to talk a little about their luciferase substrates and answer any questions you may have about them at the end of the presentation. So I'm handing it off to Simon, who's a product manager for their cell and protein analysis portfolios. So Simon.
Oh. All right. Do I need to click anything or will it go to the next slide?
I will click it. Yes.
Okay. Thank you so much. Appreciate it. Yeah, so hi everyone. My name is Simon Moe. I'm actually a product marketing manager, and so I'm primarily focused on the commercialization and marketing efforts for a lot of our reporter technologies at Promega. I really appreciate the time to talk a little bit about nanoluck luciferase. It's a small, bright reporter that hopefully by the end of this you'll be convinced is really exciting. And so if you haven't heard about it before, it's 19 kilodaltons, and it generates light just like as was introduced in the beginning of this talk through a chemical reaction with nanoluck and its substrates. So we have in vitro substrates like furimazine and some of these in vivo substrates that I'll be talking about on the next slide. When it oxidizes this, it's producing that really nice visible blue light that you can use to track a variety of biological processes. Nanoluck is a little unique because it's actually ATP independent, and so it has unique advantages for applications that traditional luciferases like Firefly or Ranilla might not. So these include exosome research, biodistribution of LNPs, as well as target engagement on cell surface receptors. And it also has really low immunogenicity, so you can be a lot more confident that you're getting responses not due to adverse immune responses or off-target effects. And so the graph on the right-hand side is to show you that it is dramatically brighter than Firefly or Ranilla, but we're not trying to push those out of the market entirely because there are distinct advantages to using Firefly and Ranilla still, but because they're using different substrates, you can actually get this really nice multiplexing between nanoluck and Firefly to do dual luciferase experiments. And then at Promega, outside of in vivo experiments, we have a whole portfolio of reporter tools to use nanoluck in cell-based assays. Partnering that with in vivo imaging, you get this really nice continuity, and you can translate your work from cell-based models into in vivo models without having to dramatically change your model. And then lastly, if you guys haven't heard of BRET, it's bioluminescence resonance energy transfer. So it's when you're using a luminescent donor to excite a fluorescent acceptor to get a lot of the advantages that were touched on earlier in this talk with fluorescent reporters. So, deeper tissue imaging, voltage and calcium sensors, and all of that is powered by the really bright light of nanoluck that's powering that fluorescence. The workflow on the bottom is a little redundant with the rest of the talk, but I still want to go through it. So again, when we're setting up these workflows, you have to introduce your luciferase reporter. You can do that a variety of ways, such as like cells, viral vectors, or transgenic animals. You're then formulating and injecting your luciferin small molecule, so your substrate, placing this into some sort of low light imaging system like the one that we've been talking about, and then getting your data processing. And so if we could jump to the next slide, we can talk about some of the substrates that have applications for in vivo imaging. And so the first one is fluoropyrimazine, which is specifically designed for in vivo detection of nanoluck or reconstituted nanobit, which is specifically designed for like protein-protein interactions. And so FFZ was specifically designed for tracking gene expression, cell growth, migration, other biological events, really whole system-wide applications for luminescent detection. It works really well for both IP and IV injections, as you can see in the top right. These are mammary gland, growing tissues, and so you can see some really nice signal from those. And so system-wide FFZ is really powerful and has a lot of advantages. But when it comes to measuring, the central nervous system or the brain, it's challenging because we have this blood-brain barrier that makes it hard for these substrates to, permeate through. And so we actually recently developed a substrate specifically for crossing that blood-brain barrier, which is cephalopyrimidine on the bottom side. And so if we look at some of these images in the bottom right-hand corner, we can compare forebrain reporters, so CAMK2 and VGAT, and you can see that CFZ actually has really nice localized bright expression with these reporters genes inside of the forebrain, compared to FZ, which is furimizine, the in vitro, and then fluorofurimizine, which is the one up top. D-luciferin is the firefly substrate. And so again, CFZ is really kind of designed for that specific central nervous system experiments. And you can see that it generates a lot of light. But, when we're thinking about these like deep tissue imaging, this is generating light at about 460 nanometers. And so it's not ideal for like deep tissue imaging. And so we've seen some really interesting data where people are pairing it with something like Antares, which is like a light modifying version of this, so that you can actually get the wavelength a little bit more appropriate for some of that deep tissue imaging, as well as you're getting a signal that's so bright, you can actually get real-time video rate imaging with a luminescent reporter. So you can see live animals moving around through bioluminescence, which is pretty unbelievable if you think about like where luminescent reporters were a decade or two ago. And you can also do some really nice calcium flow imaging with CFZ, again, because you're looking at that brain imaging. And so this is done with CAMBI, which is a nanolect calcium sensor. And so, yeah, we're really excited about these substrates. We're really excited about kind of the future direction of nanolect. I think it pairs really well with a lot of the work that I'm assuming or hoping a lot of you guys are doing based on the way that the rest of this talk went. And I really appreciate your time, and I'm happy to answer some questions with the other people from Promega that are on the call. Thank you.
So training and future directions. So because there is an X-ray component to the new system, X-ray safety training is required regardless of you're using the X-ray or not using the X-ray. And so that will need to be completed prior to in-person training sessions. And I will be hosting those after the webinar. So please send me an email if you're interested.
Great, thank you, Rita. I do want to continue on here on this slide to give everybody a heads up about a STARR LEGO user working group that is in the works, that is being established for the optical imaging community. And I welcome you all to join once we do make an official announcement early next year. It will bring together common Lago-X and even Xenogen users from the past to really share their ideas, their project, and even having a troubleshooting discussion and a conversation, perhaps once a month, held virtually, to really build a networking community for this equipment. So keep an eye out for this launch of the user working group. Hopefully you all can join in that discussion in the near future. So we're very excited to announce that today. Okay. Slide. Oh. Can advance the next slide, perhaps. There you go. Thank you. So you can get in touch with myself as well as Rita. Our email contacts and information is noted here on the slide. For staff support in terms of instrument training, software training, obviously get in touch with Rita, and systems maintenance for the Lago-X. We've also noted the vendor support. Bruker Spectral were also on the call as well for any additional technical support as needed for any hardware software issues, obviously that will go through STARR. But this is just more for our user awareness. We've got Scott Ireland and Mike Rule as well, the application scientist from Bruker for additional support as needed. Next slide. Thank you. And lastly, to end our webinar this morning, I do want to acknowledge the tremendous support from Bruker Spectral Instruments for the equipment installation over the last month or so here at STARR. And also, I do like to acknowledge the UHN procurement team, our facilities team here at UHN, as well as our green team, and as also our research innovation course team, who has always given tremendous support in hosting these webinars. So thank you for that. And most importantly, I do want to highlight the purchase of this equipment was supported by our Canadian Foundation for Innovation, our CFI funding, for making this possible. And our STARR contact information, for those who don't know, we do have our homepage, our LinkedIn, and our phone and email contact. And please don't hesitate to reach out to us, myself or Rita, for any additional questions that you may have that may not have been addressed, that may not be addressed today. So we are now going to open up the platform for any questions that you may have, any comments. Post them in the chat, or you can turn on your mic to have them answered here. We look forward to hearing what your thoughts are on the Lago-X. So we have our first hand here, Marguerite. You can unmute.
Okay. Yeah, thank you, Rita and Nas, for the great presentation. I have one comment, and then I have a couple of questions for I think Simon from Promega. So first, you mentioned that you can image up to 10 mice in one setting. I think we should let the user know if they require the injection of luciferase, you need a couple extra hands to make sure they all get the injection pretty much at the same time point, and to fit the curve, the curve you presented, to get the optimal imaging time point. So that's very important before the people get too excited and one person try to image 10 mice at the same time. Then I have a question for, I think, Simon from Promega, about the NanoLook. How are the costs compared to the conventional D-luciferase?
Sure, yeah, it's a good question. So the cost is a little bit more than D-luciferase. Firefly and D-luciferase have been in the market for a really long time, and so I think it's a little bit more competitive price-wise there. But again, I think that NanoLook isn't really trying to push those out of the market. It's really trying to position for applications that Firefly might not be optimal for. And so, because of that, and because of the bright signal, you can get, expression down to a little bit more physiological levels. And so, because of that, it's kind of positioned aggressively there. But, obviously you can talk to your sales rep, and you can have those kind of conversations when appropriate with them to see if you can negotiate with pricing. So
thank you.
Mm-hmm.
Any questions or comments?
I think there's a question in the chat that was posted. So the question was asking, how broad is the bandwidth for each of the LED excitation wavelengths present in the Lago-X system? Is there significant overlap between the distinct excitation wavelengths?
Rita, over to you.
From my understanding, it's actually quite precise that the LEDs excite at. In terms of like how far away they are from each other, it's about 30 nanometers.
Okay.
I don't know if like Scott or Michael have anything to add to that.
So Scott, I believe Scott's online here as well from Bruker Spectral. Any comments from your end, or Michael?
Yes, and I see that Melanie Smith has also responded in the chat to that the LEDs have a 20 nanometer bandwidth, so it's 10 nanometers on either side of the described wavelength.
Mm-hmm. Thank you, Scott. And yes, I did see Melanie's response there.
Yeah, I had a question as well too. Also, it was great to see that comparison slide you had comparing between the IVIS and the Lago-X, which is pretty useful to see. Just a general question I had just from listening along was which modality generally produces better image quality, bioluminescence or fluorescence?
So I wouldn't really say that one is necessarily better than the other. It really depends on your applications and what you're looking for, because both modalities have their share of pros and cons. But if you're looking at multiple targets, fluorescence kind of allows for multiplexing, whereas bioluminescence does not. But bioluminescence has a higher signal to noise ratio, so it makes it better for in vivo optical imaging. So you minimize your off-target sites, or you get less non-targeted specific signal.
If I may also add, Rita, as well, to that point, I think it also really depends on the location of the tumor. Subcutaneous, I think those signals are fairly straightforward and more obviously visible in that regard. If you're looking at more orthotopic models, and I know that one of the models that was presented here, the cervix orthotopic models, which is the work that I'm doing with Michael Milosevic and some of the word research that I have ongoing, and we did do a sort of a practice run on that system on the Lago-X, where the tumor was implanted or grafted on the cervix. So it is an orthotopic tumor. And in DS red fluorescence, and so it was a bit of a challenge in terms of being able to see some of the that signal without having to open up the peritoneum and so forth. So the depth of the tumor above the muscle fat and so forth sometimes does pose a challenge, and so hence location matters in terms of your accuracy of your signal. And even so, just to add further on, is like the lymph nodes as well. That was also noted in that slide. The lymph nodes, we're looking at like 2 millimeters or so forth. But yeah, we were able to see that, weren't we, Rita? With obviously once we opened up that tumor, and we were able to see the lymph nodes in the aortic chain bright very, very nicely there. So it's all it's a depth issue as well that can be quite complicated on that regard, depending on your study and the tumor model and where it's located.
I also had a question about if I have cells that are labeled with a fluorophore and they also express luciferase, could I image both at the same time, and would there be any overlap in those signals?
It's a great question.
Yes, thank you for your question. You could image them both at the same time using the system. I'm not too sure if you can actually overlay them on top of each other. I think that's like a Michael or Scott question. But depending on the fluorophore that you chose, there could be some potential overlap between the fluorescent and bioluminescent signals. And so using like a redshifted luciferase might help minimize it. But for best practice, you should do the fluorescence imaging first before your bioluminescent imaging, or like before you inject for bioluminescence. So then the signals are quite separate. They also have like an unmixing protocol in the software, but I'm not too sure if that applies for bioluminescence and fluorescence signals together.
It's primarily designed for multicolored fluorescence, where you're using multiple probes.
Yeah.
There have been some people who've used multicolor bioluminescent tags as well and used similar technology. And there's absolutely no problem with doing both in the same experiment. You just have to build a protocol to take those set of images.
Mhm.
Another question then, what energy is the X-ray and general application for it?
So the system has two X-ray energies, either high or low. The high energy is 50 keV and the low energy is 10 keV. General application, it helps to provide a lot of anatomical information, helps to essentially better pinpoint where your signal is coming from. More of like a confirmation, I suppose.
And to build on that question,
maybe I can add to this. I work with bone metastasis model, so I'm very excited to be able to get a very precise location of the bone metastasis with the bioluminescent tumor cells. So unfortunately, when we had the last set of rats around, the Lago-X was still standing in the hallway. So we're injecting rats again. So by the end of the month or beginning of December, we may have some images available where you can see the tumor in the X-ray and then get a good idea how useful it may be to use.
It sounds exciting, Margaret. Yes, we look forward to seeing some of those images and having you put them on the Lago-X, and let's see. That's great.
also just curious, what would the high and low X-ray energies be used for?
So the high energy X-rays are typically used for like the mice and the rats, essentially like denser specimens that you would need a higher energy X-ray to discern differences between the tissues. The lower energy X-rays are for things with lower density, such as like neonate mice or like insects, things that would need like a softer X-ray energy for. But yeah, namely just less dense things compared to the high energy, which is for higher density things.
And then last question for me, I think you might have touched on it, but just on the troubleshooting slide, I think you talked about power cycling. Can you just speak more about that?
So power cycling essentially means to like turn off the entire system as well as the computer. And then you kind of just restart it. So you'd like wait a minute and then you would turn on the Lago-X first, so the system, and then you would turn on the PC afterwards. So you know that they're ready to be like paired up. But yeah, that's what I meant by power cycle. Just turning it off and then turning it on.
Cool, thanks.
Thank you.
Are there any additional comments, questions, clarifications? Put them in the chat, and you're welcome to email, as I say, you're welcome to email myself and Rita if there's anything else that comes up after this webinar. But we're very excited to officially launch our Lago-X here at STARR, and you know, it's taken you know a while to get here, and so we're, as I say, we're very excited to get this new system up and going and get all of you to be on board on starting your new projects here at STARR as well. So we can happy to reach out to Rita to get the training and so forth, and we'll walk you through that process in terms of getting your projects up and going here. We will have Xenogen still at STARR, so eventually we will be transitioning all of our Xenogen users over to the new Lago-X. got.
Yeah, I just wanted to point out for with regards to the software that the ARRA software is able to open the Xenogen files. So if you want to be able to continue to analyze those in this freely distributable software that everybody could have, it is possible.
That's great. Got it. That's good to know. I think that if there aren't any other questions, I don't think I see anything else in the chat there. We can definitely end. Thank you, everybody, for joining our STARR webinar. As I say, we're very excited to launch our new Lago-X system here at STARR. Please don't hesitate to reach out to myself and Rita, and we'll help you get started on our new system. Thank you all.